in our own group, we try to look for, again, broad spectrum antiviral target. To shift the gear a little bit, we try to look a little bit further at this question. Is there really a way to have some method to really give you a broad spectrum antiviral effect? Actually, in our nature, bat, the only flying mammal, really seem to have found the solution to that problem. Bats are well-known reservoir for many pathogenic viruses, including SARS, Ebola, and MERS, and it's believed to be a reservoir for the COVID-19 viruses in this epidemic. However, bats can tolerate these pathogenic viruses without obvious symptoms. Previous genomic evidence have pointed to that bats have really a highly evolved innate immune system. So we try to do, as I mentioned, check the bat cells and see is there any gene that really can important for this virus tolerance in bat. So we used the genomic tool called CRISPR-Cas9 system. Basically allows you to interrogate one gene at a time, to knock out a gene at a time and see what's the effect knockout on virus infection in bat cells. Here we developed our basically a genetic screening system to check one gene at a time in bat cells to see what genes might be very important for the virus, for the bat tolerate this bat cell tolerate the virus infection. We actually identified about a dozen or so cell genes, and we followed up on one of the gene called the MTHFR1, which is an enzyme also involved in nucleotide synthesis. As I mentioned, just like DHODH, it's an enzyme involved in nucleotide synthesis. And since for this gene, we have small molecule inhibitors. Then the rationale is that if you knock down this gene, the virus can no longer kill can have much lower infectivity to the cells. Then you can use a small molecule to do the same thing to inhibit viral infection in cells. Indeed, we found that the inhibitor of this gene called carolectin, they can potently inhibit virus infection, different kinds of viruses. Here, I'm showing a Zika virus as well as mumps virus are inhibited by this inhibitor. Those are in bat cells. We also show that in monkey cells that this inhibitor can also potently block the infection of COVID-19 or SARS-CoV-2 of monkey cells. So this is give you an example that by searching for new host factors important for viral infection, we can identify new viral targets, antiviral drug target, as well as new idea how to contain virus infection. So in summary of the whole lecture, I hope I can convey you the idea that the virus infection of humans, we depend on hundreds of our own genes to receive or to enable the virus infection. Those host factors really are potential target to develop new antiviral drugs. Our human cells also developed both innate and adaptive immune responses to contain the viruses. This includes phagocytosis, interferon, NK cells in the category, under the category of innate defense, and T cell responses and B cell responses in the category of adaptive immune responses. And we can develop antiviral drugs based on this understanding of the viral dependence and our immune responses, as I give you a few examples just now. So in the end, I want to emphasize that the idea that we studying the virus whole interaction, we hope to develop broad spectrum antiviral drugs that might prepare us not just for COVID-19, but also for some virus we don't yet know, some that might emerge in the future, which is really important to prevent another outbreak like COVID-19. And thank you for your attention. And if you have any question, you can send me an email. I'll try my best to address them as soon as possible.